Okay, here it is. Great. So I think we can start.
Okay, great. So let me know if you have any technical problems hearing me or anything. So thanks, Giovanni, for the invitation. And like he said, yes, so far in the past, I've been happy to contribute the EDS and FIST modules in PLUMED. And today I'll talk about FIST, which is something that was done with my own group. And I'll mention some other work at the end that we did recently with PLUMED that is related, but not about FIST. So before I get started, I just want to acknowledge my group and funding, and in particular for this talk, the FIST project was done with Eric van den Einden, who's a math professor here at NYU, Michael Hartman, who was a postdoc and is now at Merck, and Yuvraj Singh, who's currently a grad student at NYU still, close to graduating. And so I'm just going to give you an overview here of the motivation. So I want to study how biomolecules behave when they get pulled on by mechanical forces. And that could be because people do single molecule experiments where they attach that molecule to something where they can apply forces, or because they're pulled on by forces within the cell, like molecular motors or flow, or the cell gets squished, things like that. So just from a statistical mechanics simple perspective, we're normally interested in simulating some energy function or Hamiltonian like this H. And if, for example, it looked like a double well, which is a very common example system that we'll study in this masterclass, what happens when you apply a force along this x-axis, Q, is it tilts the energy landscape. And you can see why it would do that, because if you just take the energy and you lower it by F times wherever the position is, and then you reset the minimum to here, it would look like this. And if you put a negative F in here, then the energy would go up. So the question that we want to know is what happens though when it's not a one-dimensional potential and you have to consider all the degrees of freedom of your system? Can you sample well enough to see what would happen when these forces are really small, which are the ones that are relevant for biology? So the scale that is important in biology is a piconewton, and the thermal energy KT is around 4 piconewton nanometers. So it's really like similar to the forces you experience just from thermal fluctuations. Okay, so what kind of I expect is that if you do a one-dimensional projection of a complex system and you apply a force, it's not that it will just tilt the system, but the force has a different effect on every configuration that you average over, and so it could have weird effects. So just to give some background, as Giovanni mentioned, an earlier way of studying systems with force that's motivated by experiment is called steered molecular dynamics. And I have some information about this in the master class, but I didn't actually put an exercise on it. But we may hear more about it in a guest lecture from Guillaume Sternemann next week, or a second part to the lecture. So the idea of these original experiments was that they would attach a protein to either a bead or an AFM tip and pull. And by pulling, they would unfold the protein. And they were hoping that they could understand the folding mechanism of proteins by pulling them until they unfold. So whether or not you believe that's the case, people were really interested in doing this in simulations as well. And so some of the major work on this was done by Klaus Schulten's group, which is, here's a picture from a famous paper about that, where they studied the protein Titan in 1998. And one thing that you might notice here is that in order to unfold this protein within a normal amount of simulation time that they could do, first of all, you can see in this picture that they use this water droplet kind of, not a big periodic boundary system. And then they also had to pull what turns out to be really fast compared to experiment, with the result being that the forces you apply are very large, like 1,000 piconewtons or 2,000 piconewtons. So that's one of the reasons that mainly in my research, I'm not using steered MD except to generate initial configurations between two states, but there's still a lot you can learn from this. And one of the things you can do, and was done even in those early papers, was to use something like the Jarzynski equality to compute the free energy by looking at the fluctuations in the work that's done during the steered MD trajectory. And this is actually something that's very easy to do in PLUMED, and it might be something I add later as a bonus exercise to the masterclass, because I did set it up. I just didn't include it to not make it too long. Okay, so some famous work on how to analyze those simulations come from Gerhard Hummer and Attila Szabo, for example, in this paper. Okay, but that's when I started my group. I wanted to pull with like 2 piconewtons and see what happened. So one project that we did with Eric van den Einden is to see if we could use pulling as an enhanced sampling technique. So the goal was to see if we could get proteins or peptides to overcome energy barriers by pulling on them. So this is similar to how we would use temperature in replicate exchange, which you've, I'm sure, seen in earlier master classes. We would use force, and the idea is that by destabilizing the protein with force, maybe it will cross some energy barriers, and then we can observe more sampling. So to connect to some things you've probably learned about in other master classes and in class, you could imagine what it might be like to do parallel tempering in force if we want to understand the effect of many forces. So just to take 2 as an example, we would run a simulation with a low force and a high force added to the energy. We would run ND for a long time, like this would be Langevin dynamics. At some point, we would ask whether we can swap these replicas, and the formula for swapping would be the same as Hamiltonian replica exchange, which I think Giovanni did a masterclass about. And then if we swap, then we continue with those configurations. And eventually we would learn the effect of all of these different forces. And within each window, the configurations would be distributed in like a Boltzmann manner that depends on the force. Okay, so a similar method is called simulated tempering. And the idea of simulated tempering, which goes back to this paper from Parisi, is that you simulate at some force and then or temperature, and then you increase it, run for a while, and decrease it. And what you do is you introduce weights which promote uniform sampling of this thing that you're interested in. So for them, they were interested in sampling energy uniformly, and here we're interested in sampling the effect of all the different forces uniformly. And so it turns out then that you should swap with this weighted distribution probability. And if you want to have a random walk in forces, then the result is you should have your weights inversely proportional to this force weighted partition function. Okay, so given that, we wondered what would happen if you introduced a continuous version of this scheme. So this is something that was done by Van den Einden in a previous paper that I'll show you. So the idea is that instead of having discrete forces that you swap between, you make it an extended variable in your energy function, which has its own mass and momentum. You may have seen this just from having thermostats like the Nose-Hoover. And the idea is that we'll just have this extra variable, and then this momentum and extra variable and confining function will make you sample between negative and positive forces. And the idea in this paper, infinite switch simulated tempering, is to take the limit of this going up and down very fast, the mass of energy or mass of force in this case, very small, so that all the forces are sampled very fast. And then what happens is that this Langevin equation, that the force gets replaced by an average force. And the average force has some formula that you can calculate based on the weights. And afterwards, since you did one single simulation with this average force, you're able to reweight anything that you want to calculate using a set of observable weights that you calculate for each configuration that you saw during the simulation. So if you want to calculate like the average radius of gyration, then you would have this weight for each configuration at a given force. So you want radius of gyration at big force, you use the weights for big force. And if you want it for negative forces, you would use it for negative forces, and you'll see big or small proteins. You calculate these observable weights like with a formula like this and disease, and it turns out that you can do an iterative algorithm to estimate these on the fly, which I have which is explained in the paper. And so we implemented this as a FISST module in this paper, and that's what we're going to get some experience with today. So now I'm going to tell you about the test systems that we studied that are also the ones you're going to try, which are all contained in this plumed nest egg that I made for this paper. So there's 3 different systems and a 4th one that I included for the masterclass. So in the paper, we studied this potential. The idea is that it has, if you pull in the y direction, it has a stable state up high. And if you pull in the negative y direction, then it has 2 stable states. And this is supposed to mimic that helix problem that I mentioned earlier. So doing FISST and then reweighting, you get some things like this. At small force, you get 2 states. At high force, you get 1 state. If you only run a single simulation, you'll be with a low force, for example, you'll be trapped on one side. And but FISST equally samples both basins, and then you can reweight and make a movie like this, which is something that you'll do in the masterclass, where you can reweight to any force and see how the sampling would have been if you'd just done this perfect sampling at that temperature. Okay. The second system is one that we concocted, which is a bead spring model of a helix. So there's interactions between pairs of residues along the helix. That is just a Leonard-Jones interaction. And the point is that if you simulate with no force, the system will be stuck on one side. And if you pull upwards, it will actually, interestingly, be able to sample both parts. And then if you pull a large amount, it will only sample an extended state. When we do FISST and reweight, we see that the 2 states are actually equally populated. At intermediate force, there's force where maybe each state is equally likely, the left, right, and extended. And at high force, there's only 1 state. So you're going to test this out as well. And finally, we picked a peptide to study alanine 10, which maybe was studied in some earlier master classes. So for alanine 10, what we did is look at the end-to-end distribution and pull on the ends. So if you pull to the right or pull on the extended direction, then it gets more extended. And if you pull in the negative direction, it emphasizes some state that's at small distances. And the other thing to emphasize, which you'll do in this masterclass, is that you can reweight any quantity. So this is the one that was actually pulled on explicitly. And this is some other quantity, the Ramachandran plot averaged over the 10 residues. And if you do harder pulling, what you'll see is that this upper left corner is more populated, which corresponds to the extended state. So that's something you'll also see in a minute. So just to conclude about FISST, we found that you can calculate molecular properties with applied force, and it will actually accelerate sampling in certain cases. We derived how to implement this infinite switch limit, and the only parameter you really have to choose are the force range you want to sample and the learning rate, I guess. So you can then pick any, something to emphasize, which I also said in the master class, is you don't just have to pull on a distance, you could also pull on some abstract quantity, any CV that's in plumed. So if you want to pull on the, on NMR couplings or something, then you could do that. And currently what we're trying to do is seeing how we can combine this method with other enhanced sampling techniques and perhaps trying to see how you can optimize the initial weights to get better performance. And then just to mention one more thing that I didn't have time to put into the master class, but work from Wilmar, who's a graduate student at NYU. We published this paper this year on studying rates with applied force. And the idea is that when you pull, you lower this barrier, and so rates should change. So for example, we studied this cavity ligand model from the Byrne lab, which has slow unbinding of this ligand. And what we did is we took advantage of work from Paranello and others the method infrequent metadynamics. So what we did is we applied constant forces, created a small bias with metadynamics, which caused the molecule to escape faster. And then what you do is you get very fast escape times with the bias, like in the nanoseconds. And then you use something called an acceleration factor that's implemented in Plume to get times that are on the seconds scale. So this is something you could also do, and I may add later as a bonus to the masterclass. But it's outside of the FISST module. This is just showing that for constant forces, you can do any enhanced sampling technique you want. So lastly, let's just take it over to the documentation for this masterclass, which was posted this morning, and then I can show you what the plan is. Okay. Great. Okay, so in this masterclass Plume documentation, what you'll see is that the learning objectives are to see how to do constant force pulling in Plumed. You can learn about doing steered MD in Plumed. This part I could take out. I may add something about calculating energy surfaces, but I took it out. And how to use FISST to apply force and reweight. So what you need to do is install Plumed with GROMACS and LAMPS, get some input files from here. And I actually included some output files for one part in case you don't want to run a long alanine 10 simulation. And then I explained some of the backgrounds that I've talked about here, how steered MD works and how it's implemented in Plumed, how FISST works. And then what we do is we go over to this one-dimensional potential, which is a fourth example system. For this one-dimensional potential, which is using the PESMD simulation function of Plumed, the idea is that when you apply a constant force, it will make you more or less likely to populate this right-hand basin. In order to prove that, I picked a double well with a relatively high barrier, and then what you're going to do is run metadynamics with applied force so that you go back and forth very fast, and then you can tell what the energy surface is, even though it's a one-dimensional system, and you don't technically have to do maybe a fancy sampling to understand this effect. That's what I set up here, which I think builds nicely on some of the earlier master classes. Then we have this V-shaped potential, which I talked about in my lecture, which is implemented here in PESMD. And what you'll do is learn how to see an example of how to apply a force from -15 to 15 with FIST that you can then apply in the next examples. You just have to fill out some output files. And then there's a script to reweight those output data to make this plot, which will show you how reweighting works. And you can use that idea in the later parts of the lecture or of the masterclass. The third part is this 2-helix model, which I showed in my talk. And I have input files and a command for running this with LAMPS. And so what you'll do is you can try pulling at a single force. Maybe this file name might not be the one that's in there, but there's a plumed file for just running it with no force and one for running it with FIST that you have to fill in. So there's a missing part on actually the FIST section of this input file. And if you do run it, what you'll see is that if you take the negative log of the histogram of FIST, you'll see that both sides of this free energy are sampled evenly, whereas these little circles that you can see in the right-hand side here are from an unbiased simulation, which shows that it's trapped in this case in the right-handed helix. And then you can use FIST to reweight, and you'll see that you can either be both in these left and right helices, or for some bigger force, you can be in, you can evenly populate the 3 states, and for some even bigger force, you can be at the top. Okay, I have to add a color bar to that. And finally, I And I should just mention that I included a Python script to calculate this x-axis that you're going to reweight with, which is a bit complicated. And then finally, I have this example for alanine 10. I have output files for this if you want, and you can see how when you pull at different forces, the end-to-end distance is shifted to the right or left. And then you can also reweight this other quantity, like the I mentioned the Ramachandran plot, to see that you can reweight other quantities and that it makes physical sense that you go to this extended basin. And this is something that's also been shown in earlier papers by Guillaume Sternemann, who's going to probably talk next week. Okay, so I hope you'll check this out and give it a try and let me know if there are any problems. And then I'll be happy to talk through some of the solutions next week.
Okay, thanks, Glenn. So is there any question? Otherwise, I have a question, actually, a curiosity.
Yeah, sure.
So here you have to choose these mean force and max force values.
Yes. Mm-hmm.
And also you have to choose how many values of the force you interpolate with, right?
Yes.
So how crucial are these parameters in general? And also, so in theory, it's not even necessary that the forces are equally distributed across the range, right? So
right. So yeah, I didn't
make
mention
sense to play
mm-hmm.
with these types of ideas, like trying to have a different equilibrium distribution of forces.
Yeah, so what actually happens is, so the minimum and maximum kind of matter because it's what you want to study, but we also found that maybe you should have some, even if you want to pull really hard, you should have some negative force to kind of balance it out with some restoring to or lower force configurations, although that's just heuristic. Like technically you shouldn't need that. The interpolating forces are actually just for doing numerical integrals, and they're using some like Gauss-Bessel thing that is not evenly spaced that's supposed to be for an optimal numerical integral. And so you just need enough of them to do this integral of like W of like some W's and e to the beta f and so forth. So they're supposed to be distributed in a nice way, but the weights in the end are what really distribute the sampling. And those end up being like some very hopefully inversely proportional to this partition function, which means that they get distributed depending on how likely, you know, kind of the free energy of your system to be extended or not. Mm-hmm.
Okay, I see. So in the end, if you change this number of points, so the only thing that changes is how expensive is to compute this integral, which I guess is negligible usually, right?
Right, negligible, should be. Yeah, so it's probably better to have it on the large side, except it makes very large output files if you have a lot of columns. Yeah. The other thing is that although it makes sense to have a lot, because what I'm mostly showing here is just the force. You can kind of only then reweight to the forces that are in at these nodes of the integral, although you could calculate it for in-between ones. But I was just looking at that formula again today, and you kind of have to interpolate between the weights at the different values in order to redo the calculation. So it's better to have a lot of points and just choose the one closest to what you're interested in. And if you want to do exactly zero force, then you need like an odd number of interpolation points from negative to positive, same value.
Yeah, clearly. Yeah, so 1 point should be on the zero.
Yeah, which isn't the case for this like minus 10 to 100, but you know, it's fine. Depends what you want.
Yeah.
And then, yeah, I think the only parameter then is this learning how often you update the weights, which is mostly important at the beginning. And that's very similar to the max end module and EDS module, where you have to, or functions, I mean, where you have to basically do a continuous average over some number of steps before you.
Yeah, and I would say also with the simulated tempering, you have something like that to choose.
Mm-hmm.
Right, because you have to update your partition functions.
Mm-hmm.
But
I guess in simulated tempering, at some point you fix the weights, and then that's what you should use for your sampling. But here, according to Eric, and Ben Limecooler in the other paper, you are supposed to be able to use the entire simulation from the very beginning. That's what I've been told. And so I think that's an interesting point to mention, like it's an unbiased estimator the whole time.
I see. Even though the trajectory is out of equilibrium, strictly speaking.
Right.
Okay, I see.
So yeah, even though you're changing these weights and changing how you apply the force, it's supposed to all combine in the wash to give you the right answer.
Okay, great. So other questions, curiosities? Okay, good. So then if we have no more question, I think we can close here. And we were just discussing with Glenn, and likely next, so the solution will be next Monday, but 1 hour in advance. So at 15 Central European time, it will be.
Mm-hmm.
Okay, great. So
Great.
thanks again, Glenn, and bye-bye to everyone.
Mm-hmm.